In 1989, a genetically engineered brand of L-tryptophan, a common dietary supplement, killed 37 Americans and permanently disabled or afflicted more than 5,000 others with a potentially fatal and painful blood disorder. The manufacturer had for the first time used genetically engineered bacteria to produce this. It is believed that the bacteria somehow caused this and had become contaminated during the DNA modifying process. This example and many more are reasons why eating genetically modified foods is dangerous. The contamination of genetically engineered foods can become contaminated during the modifying process, become allergenic through the addition of foreign genes, and can increase risks of cancer. The contamination that occurs during the process of modifying the dietary supplement is not the only way contamination can occur from genetically engineered foods. Dr. Arpad Puste's research found that genetically engineered potatoes that were spliced or connected with DNA from a plant and a commonly used viral promoter, the cauliflower mosaic virus, or CAMV, damaged the vital organs and immune system of lab rats fed the genetically engineered food. This was most likely caused by the CAMV viral promoter, which is spliced into many kiwi foods and crops today. Evidence confirms that the CAMV promoter is able to transfer intact to organs and has the potential to be active in human cells. Dr. Joseph Cummins, a professor in genetics, states that it has been shown in the laboratory that genetic recombination will create highly virulent new viruses from such constructions. He also goes on to say that the widely used cauliflower mosaic virus is a potentially dangerous gene and is a pararetrovirus, meaning that it multiplies by making DNA from RNA messages and is very similar to the hepatitis B virus and the HIV virus. Furthermore, the studies of Dr. Irina Yermakova at the Institute of Higher Nervous Activity and Neurophysiology of the Russian Academy of Sciences showed that the mortality rate for newborn rates was 6 times higher than those not fed the diet of genetically modified soya, which also contained the CAMV promoter. 6 times as many were also severely underweight. The study also indicates that the CAMV promoter is capable of promoting gene expression in human cells and suggests that women who eat genetically modified foods while pregnant risk endangering their unborn babies. More scientists around the world are warning that genetic manipulation can increase the levels of natural plant toxins in food or create entirely new toxins in unexpected ways by turning on genes that produce poisons. Genetically engineered crops can also be contaminated in a way that indirectly promotes the development of antibiotic resistance, which is difficult or impossible to treat common human diseases. When gene engineers splice a foreign gene, or called an antibiotic-resistant marker gene, or it's linked to the gene, some researchers warn that these genes can unexpectedly recombine with disease-causing bacteria in the guts of animals or people who eat genetically engineered food, contributing to the growing public health danger of antibiotic resistance to infections that cannot be cured with traditional antibiotics. Examples include new strains of Salmonella, E. coli, Campylobacter, and Enterococci. Besides becoming contaminated, foods can also become allergenic through the addition of foreign genes in the modifying process. Since humans have never before eaten most of the foreign proteins that are now being spliced into the foods, they have the ability to create unexpected allergic reactions. According to toxicology.org, although testing strategies are currently being used, no single test is fully predictive of human responses. As British scientist Dr. Miwan Ho points out, there is no way to predict the allergenic potential of GE foods. Allergic reactions typically occur only sometime after the subject is sensitized by initial exposure to the allergen. On top of contamination-related harm and allergic reactions, genetically modified food can also increase the risk of cancer. The genetically engineered recombinant bovine growth hormone, or RBGH, which is a hormone used to make cows produce more milk, is currently used today, even though scientists warn that significantly higher levels of insulin-like growth factor in the milk and dairy products of injected cows could pose serious hazards for human breast, prostate, and colon cancer. A number of studies have shown that humans with elevated levels of this insulin-like growth factor in their bodies are much more likely to get cancer. Significant infiltration of RBGH in the prostate of rats as well as thyroid cysts also indicate potential cancer hazards from the drug. Although no other industrialized country has legalized its use, the U.S. still allows the use of RBGH in their cows. Even the GATT Codex Alimentarius, a United Nations food standards body, has refused to certify that RBGH is safe. On top of cancer concerns, the use of RBGH also increases illness and the need for antibiotics in cows, which also can lead to an increase in antibiotic resistance in humans. In conclusion, the genetic modifying process can contaminate food, such as the use of the cauliflower mosaic virus, which has the potential to cause illness, can make food allergenic through the use of foreign genes, and may increase cancer risks through the use of hormones such as RBGH and insulin-like growth factor. Therefore, it can be said that the consumption of genetically modified foods is dangerous.
All right, the proposition is very clearly identified. There are supporting points that pop up. I think that you need to identify them as the structure for the speech in the preview. It really seems like you just start talking about things, and part of the problem is that it feels quite a bit like you're rushing into the presentation and then rushing to get done. So you need to pace yourself a little bit more. However, the signposting was relatively clear. I think you could use some numerical signposting on those points because the second point seemed to have 2 or 3 parts to it, and I wanted to make sure that I was all in the same place. And sometimes I felt like I was actually on to a third supporting point, and listening to you when you finally signposted that last point, it was clear that it was like a second or a third element on that middle point. So that's where some additional signposting would help a little bit. I thought that you did a nice job giving us some examples. The explanations, I think, are a little bit rushed. You did a very good job citing sources on your information. At the end of the speech, you had a pretty effective summary of where you were trying to go and how you got there. And I think maybe your argument would be a little more focused if you, for instance, took one of those claims and focused on it a little bit more. The bovine growth hormone, for instance, is one of those places where there's a lot of controversy and a lot of information, and that could have been a speech by itself. And that's part of the problem, that you're trying to get so much in here that it feels a little bit stuffed. And this would be one of those places where some editing and maybe a more judicious focus would work better for you. All right. Thank you.